Hello friends, yeah, welcome back. Today we're going to talk about tracing the source of a biological agent. We've been talking about bioterrorism, biocrimes, and all of that. So one look at what do scientists do, to trace the source. We've done a video on what scientists do to identify the organism, which is important. You need to know what you are dealing with. What kind of organism is it that you are dealing with? Is it a virus? Is it a bacteria? Is it a parasite? What kind of organism are you dealing with? You need to know. Is it fungi? You need to establish what it is before you begin to know the kind of response. Now we are going to look at how do we trace the origin. If we're trying to trace the origin and the source of this infection, how do we do it? What are some of the things that we do? Number one, some of the things that scientists do is what we call strain level identification. Strain level identification. Now, what does that mean? Now, in general biology, we know that when we do taxonomy, when we classify organisms, put them in groups and classes, we use your phylum, your kingdom, phylum, genera, species, family, class, and all of that. Good. So at the end or towards the end of the classification, we have genus and species. So you say Panthera leo, that is your lion. So Panthera is the genus, Leo is the species name. That's good, and that works for biology. But in microbiology, we go a step further, and there are what we refer to as strains. The meaning of strains is that we realize that when you have organisms, you can have organisms that bear the same name but have slight differences. They have differences in their genetic makeup. In fact, you can have organisms that have the same name, and some of them are able to cause disease, and some are not able to cause the disease. We say one is virulent, the other is avirulent. Simple example, your Streptococcus pneumoniae. So you have virulent form, you have avirulent form. So what are we saying in essence? We are saying that for bioterrorism and in bioterrorism investigations, many times you have to look at the organism and be able to identify it all the way to the strain. So if you say that, for an example, you suspect that Bacillus anthracis is being used as a bioweapon, as a bioagent, as a scientist, you want to go to strain level identification, meaning that you want to identify it all the way to the strain to determine what kind of strain of your Bacillus anthracis is being used for this attack. And why is strain level identification important? Strain level identification is important because we are able to now do we are able to now draw what we call phylogenetic trees, meaning that we are able to look at the evolution of this particular organism. And why is that important? It's important because we can use that to trace the source of the organism. We can use that to try to find out which area or which places do we have this organism being more prevalent in. And if we find that out, then we're able to push further, and try to identify the source, try to identify the place where this bacteria may have originated from and where it is now and how it is now being used as a bioweapon. And this can help the authorities, it can help the police and other agencies of government to begin to do tracing, try to find out who maybe may have come in from that area or may have come from that area or may have flown in from that area. So that way we're able to identify the origin and source of this infection. This is very true. Another thing that we do is toxin detection. Now, like I told you before, that when a bioagent is used for a crime, it is not always the agent itself, the organism itself that is being used for this crime. Many a times, it may be the spores of the organism, like I said before. It may be other metabolites, proteins, other metabolites of the organism, and sometimes if it could be toxins of the organism that is being used. Simple example, if a case of food poisoning occurs and the authorities or scientists are trying to identify, that there are organisms that have been implicated in food poisoning. A simple example is your Clostridium botulinum, and it has the toxin that we call botulinum toxin or Botox. So what we are saying is that sometimes we use the product of the organism, not the organism itself. So maybe it's toxin, you get its byproduct, any product of its metabolism, which we now find out that that product of metabolism can be used to commit a crime. So we are saying that we can try to detect toxin to see if a particular toxin has been used for a biocrime. There are assays, there are biochemical assays that can be used to test what kind of toxin is this toxin. And then from there, we're able to, generate inferences. And, if we're able to find out the kind of toxin that is being used to commit a biocrime, that means that we'll be able to trace it back to the kind of organism that is being used. And that can help us, to be able to find out, to trace the origin of these bioweapons. Remember that all we are doing in forensic microbiology, we're trying to help the agencies to be able to, find out, solve crimes, know what it is that caused or who it is that caused a particular crime. Now, also there's what we call genetic fingerprinting. And it's important to know that every microorganism, even humans, every organism has a unique genetic fingerprint. And what is this genetic fingerprint? It's really large. The discussion of this is really large. But let's narrow it down to some concepts. One, there's what we call whole genome sequencing. Now, what's whole genome sequencing? When you talk about whole genome sequencing, you're talking about identifying the totality of the genetic composition that you have within a cell. So if you have a cell now, for an example, we're microbiologists, let's use a microbial cell, a bacterial cell. So if you have a bacterial cell, all the genetic composition within that bacterial cell, you want to know. So you want to look at, and we as microbiologists, we know that we have plasmids, we know that we have chromosomal DNA. So everything that you have within that cell that is of interest to you, and when you sequence everything out, what do you mean by sequencing? We are saying when you find out the individual sequences, you know that when you're talking about your genome now, you're talking about DNA. And when you're talking about DNA, the DNA has building blocks, and those building blocks are called nucleotides. So you know that for your DNA, you have adenine, cytosine, guanine, thymine, 4 of them, building blocks. Now you know that your DNA is a double helical structure, and you know that you can separate it into single strands. And you know that across the length of each strand, you have those building blocks, adenine, thymine, cytosine, guanine, like that, in sequence, in their millions. And so the ability to identify all those sequences in all the genetic material that is within a particular cell, those plasmids and chromosomal DNA, is what we refer to as whole genome sequencing. And we do that a lot in forensic microbiology. It's also important that we realize that there are something called SNPs or SNPs and SNVs, single nucleotide polymorphism or single nucleotide variants. Now, these are unique identifiers along the length of a sequence. So when you have a sequence of a DNA, long sequence that you have different building blocks along that length of that sequence, you can have unique points, unique sequences that keep repeating themselves, and we can use them to uniquely identify that organism. That's what we call your single nucleotide polymorphisms or single nucleotide variants. And when you have them across or along the length of a sequence, you can use it to identify that organism, and you can use it to identify other organisms that are like that organism. So all these kind of things are what we do. So that's genetic fingerprinting, both whole genome sequencing, and then when you use your SNPs and SNVs. So these are all important things to note in our forensic microbiology or bioterrorism investigation. Lastly, we do environmental samplings. So if a terror attack occurs, or if something occurs now and we're trying to identify a bioweapon, it's important for us to know, take environmental samples. So when we take environmental samples, so if it occurs in an area now, you want to take air samples, you want to take water samples, you want to take soil samples, you want to take all sorts of these samples and study to try. And the idea is you're trying to trace the origin of this organism of interest. So this is important for us to do so that guides our investigation and helps us, so that we don't miss anything. We take everything together in our investigation. Thank you for listening to this video. I do hope that you follow these things. We're here to make your science easier, to make you understand it. It's not that difficult. What we need is a basic understanding of the science, and that will help us to go further in our science. Thank you for listening, and remember to like, share the content, and do have a wonderful day.